The question of how memories are stored in the human brain is an age-old puzzle. With diseases that affect memory posing a huge threat to public health, there is arguably a greater need than ever before to unlock the secrets of how our brains hold on to information. In the 1890s, Santiago Ramon y Cajal was the first to suggest that synapses might be the key to memory function. Cajal used a technique developed by Italian scientist Camillo Golgi to produce unbelievably detailed images of brain tissue. Other experiments, including those of Canadian-American neurosurgeon Wilder Penfield, helped the next generation of scientists figure out where in the brain to look for memory storage. Penfield pioneered a new way of treating epilepsy, where he would open up a patient's skull to expose their brains and, while they were still awake, probe the brain with an electrode to determine which tissues were causing the seizures. Penfield noted that stimulating certain parts of the brain triggered memories in his patients. Eventually, there was one brain region that would come to be thought of as the most linked to memory, and to understand that, you have to know the story of patient HM. As a child, HM was involved in an accident that caused him to experience seizures, which became debilitating by his late 20s. As a last resort, HM underwent drastic surgery to remove part of his brain. Whilst this procedure did manage to control his seizures, there was one very notable side effect: amnesia. Evocatively, HM described his condition as like waking from a dream. Every day is alone in itself. It turned out that the part of HM's brain that had been removed included the hippocampus. Buried deep inside the brain, this structure had since been considered the cornerstones of learning and recollection. And this discovery is all thanks to the legacy of patients like HM. We now know, however, that no one part of the brain is responsible for everything to do with memory. For example, a recent study revealed that the cerebellum helps us store memories associated with strong emotions. However, what all this still doesn't address is how the memories are formed in the first place. Synapses are the connections between neurons. Information crosses a synapse through three main steps. First, chemical neurotransmitters are released into a synaptic cleft. These bind to receptors on the neuron at the opposite side of the cleft. This binding triggers the opening of ion channels, which are what allows electrical current to flow. One theory of memory holds that persistent changes in the strength of these connections over time allow the brain to store information. When this effect happens in a long-lasting way, it is called long-term potentiation, also known as LTP. LTP can be achieved either when there are more neurotransmitter molecules released, or when there are more receptors available for them to bind to. Either way, more electrical current is going to get through the ion channels, making the synaptic connection stronger. Over the years, however, many scientists have suggested that whilst LTP is involved in memory formation, memory engrams are necessary for long-term storage. Memory engrams were thought to have been a collection of cells that have undergone lasting chemical or physical changes, and which, when reactivated, allow for the recall of a specific memory. A series of studies provided compelling evidence for the existence of so-called silent engrams. The researchers were able to artificially retrieve memories of events that were stored in the brains of mice with retrograde amnesia. One of the main conclusions of the study was that a specific memory is stored in a specific pattern of connectivity between engram cell assembles that lie along an anatomical pathway. And in 2021, a brand new theory arose in which the human brain was likened to a computer. It's called MeshCODE, as this computer-like machinery works like biological code, where an array of biological switches alter the structural states of protein called talent. In this biological code, the talent is equivalent to the ones and zeros of digital code. When we spoke to scientist Ben Galt about this theory, he said, "With our new analysis, we suddenly realized a new discovery, that these switches are introducing a large increase in the length of these molecules. The 1 state is approximately 10 times longer than the 0 state. Since one talent molecule has 13 switches along its length, this means that the protein could theoretically be stretched out to nearly 1 micron. This idea has led to further work on the potential role of talent, plus other proteins that form the eponymous meshwork of proteins at each synapse. The work has led to some intriguing theories about memory loss in diseases like Alzheimer's. And, back in 2018, scientists achieved something that still sounds unbelievable today. They transplanted memories from one snail into another. by injecting snails with RNA from other snails that had been sensitized was enough to make them exhibit the same behavior. The study author proposed that long-term memory may not be held in synaptic connections, but was rather encoded in a series of modifications to an animal's DNA known as epigenetic alterations. The results of the snail experiment certainly seem to support this view. The author was quoted saying, "If memories were stored at synapses, there is no way our experiment would have worked." But as well as trying to understand exactly how memories work, many also ask the question as to whether there is a finite number of memories one can hold. Another thing brains and computers have in common is memory storage. In fact, there are theories about how our brains store memories that look more like computer than modern computers do. One widely cited estimate for memory storage was published in 2015. Using synaptic strength as a proxy for information storage, scientists calculated that the brain can store 4.7 bits of information at each synapse, whilst another study put the total capacity at somewhere between 4.1 and 4.59 bits. However, the approximately 86 billion neurons in the brain are not enough to fully explain its memory capacity, so therefore there must be some other cells doing some of the work. And whilst the brain's capacity is technically finite, its size is so great that it's functionally infinite. Our memory is meant to be adaptable and fluid. We are evolutionarily adapted to make distinctions of what's important and what's unimportant, and when we lose track of what's important and unimportant, that's the decline of memory. Our memories evolved to optimize us for an early Homo sapien hunter-gatherer lifestyle. In that ancient environment, it was vital to our survival to recognize recurring patterns, to correctly separate friends from foes within the natural world. But in modern life, we're exposed to so much more information on a minute-by-minute basis than our ancestors would have ever had to cope with, and it may be becoming more than our memories can handle. Clinical Associate Professor of Life Science at New York University, Nikolai Kukushkin, believes that it's not a lack of absolute capacity that's the problem. Rather, it's the rate at which our modern information ecosystem requires us to overwrite our old memories with new ones. That means things like our 24-hour rolling news cycle and constant doom scrolling through short-form social media content. Kukushkin would like to see more of a focus on what he terms mental hygiene, where people would be as mindful about what they're exposing their brains to as the food that they're putting into their bodies. Overall, though, this is an exciting time in the field of memory research. Modern technological advances mean that theories and ideas that have been postulated since the earliest days of neuroscience can now be tested experimentally in ways that have not been possible before. Much of the functioning of the human brain remains elusive. However, as long as there are new theories being proposed and older ones remembered, we will continue to edge towards a greater understanding of our minds and of ourselves. Thank you for watching IFL Science. Please subscribe for more videos like this.